This video lecture is going to cover um, the gamete development and the follicle development within the female reproductive system. And so when we take a look at the formation of the eggs inside, we're going to see how gametogenesis, which we talked about back in the male reproductive system, is the general formation of our gametes through meiosis. We're going to see a similar process in the male. We looked at spermatogenesis, and that's where we saw a spermatogonia, the stem cell, giving rise to primary spermatocyte. They divided to become secondary spermatocytes, spermatids, and then those morphed into the spermatozoa. Quantity means you make lots, make lots, make lots. This is going to be different and yet similar. And we start out with our stem cells, and stem cells, each with 46 chromosomes, are called oogonia. Now, these stem cells are going to divide while the female is still a fetus in the womb, and then they will all transform into primary oocytes. And at that point, they will go into what is referred to as arrested development. And so that means that they will hit pause. And so when you were a kid and you played red light, green light, suddenly all these eggs were given a red light and they all stop in their development. They don't divide anymore, they don't grow anymore. And so generally speaking, the female is born with about 3,500 eggs between the two ovaries combined. And that's all the eggs she's going to have in her entire life, because when she's born, they're already converted into primary oocytes. So she no longer has any of these stem cells. Once she reaches reproductive age, each menstrual cycle, about 6 to 10 of those primary oocytes will leave arrested development. They'll continue through development, and then eventually one of those is going to survive long enough to ovulate and leave the ovary. And when it does, the ovulation event is the trigger that's going to divide our primary oocyte into a secondary oocyte and a tiny little cell called a polar body. And so at ovulation, division of the primary oocyte is not equal like we saw between our primary spermatocyte into two secondary spermatocytes. Instead, we divide in order to get rid of the excess DNA. Remember, a major function of meiosis was that reduction division. We're reducing the amount of DNA as we divide the cells. So that's the job of the polar body. All the organelles, all the cytoplasm, all that stays in our secondary oocyte. And so the polar body will be this little structure that just simply dies and floats off and just gets destroyed. It has no value after that. This job is done. The secondary oocyte, this is the one that's going to travel into the fallopian tube, head towards the ampulla. If it does not get fertilized, it will never finish oogenesis, all of this development. It'll stay a secondary oocyte. It'll live for about 24 hours and not fertilize. It'll simply die off, break apart, phagocytes will chew it up. Sperm contact, however, is going to trigger that secondary oocyte to finish meiosis development, divide one more time to come up with our mature egg called an ovum and another little polar body. And so when the sperm makes physical contact with the secondary oocyte, it'll divide off to become an ovum and another little polar body. We now have all the rest of the DNA out. This is now a true haploid cell. That sperm will then enter the ovum. The two little nuclei will fuse, and this ovum will then become a zygote, a fertilized egg. So its life as an ovum is going to be like, a second, and maybe that's it. So it's going to go from secondary oocyte, physical contact, divide, fertilization, done. And so part of that quality issue is saving these steps until the last possible moment so we don't throw away energy or effort. So primary oocytes are staying in the ovary. they will have 6 to 10 of those activated every month. And then one of those survives long enough to release from the ovary ovulation, divides out to become a secondary oocyte. That floats down when it knows that sperm are in the area. That'll trigger it to divide into an ovum, which can then be fertilized. And so there's everything I just babbled about. There's the picture that shows our oogonia, which are dividing off into and becoming primary oocytes before birth. And then at birth, that primary oocyte is going to get activated, start growing bigger, bigger, bigger. And then it's going to ovulate. When it ovulates, it's going to divide into a secondary oocyte and a little polar body. That secondary oocyte will travel down through the fallopian tube. If it meets, if it does not get fertilized, it simply dies off. If it does get fertilized, it'll divide off into an ovum and a second polar body. And then that ovum will be immediately fertilized by the sperm and make a zygote. And then we're off and running as far as development. Over on the right, we're going to get into the follicle development, which we'll describe in a moment. And so that's where you see this large egg, this large primary oocyte surrounded by all these little supporting cells. You can see the relative size of that oocyte compared to all these little dots. Those are individual cells. That oocyte is one big egg, one big cell. And so it's got to have a lot of cytoplasmic material because when that thing is ovulated and fertilized, it's about a 7-day float trip from the ovary to the uterus. So it needs all that fuel to be able to survive the trip, start dividing, getting ready. So by the time it implants in the uterus, it's moving along developmentally. So it's a big, big cell. All right, that's the oogenesis. That's the formation of the eggs. So again, here we see these little oocytes in arrested development. And then here's a couple of those oocytes that have been activated, starting to grow bigger. And then here's a large mature one. You see how big it is compared to the other cells within the ovary. These are supporting cells called granulosa cells. And so you can see the relative size. All right. Now, with that, I want to get into talking about follicle development. Now, with follicle development, I'm going to look at my primary oocyte. Because when we had our oogonia before birth, and then we have our primary oocyte still in the ovary, and then we become a secondary after we leave. So whenever you see an egg is the general term, whenever you see an egg anywhere in the ovary, it's always a primary oocyte. Always, 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 always. However, surrounding it will be a number of secretory cells, supporting cells, and these cells will start to form a ring around it. They will provide nutritional support, hormonal support, and that combination of the oocyte and the surrounding cells is a follicle. And so we're going to take a look at the various follicle stages and show you how this progresses through each menstrual cycle. We start out with a primordial follicle. A primordial follicle is a primary oocyte in arrested development. The primordial follicle is a primary oocyte in arrested development. So those 3,500 eggs waiting in the wings, waiting their turn, those are all primordial follicles. Each menstrual cycle, about 6 to 10 of these will then be activated and they will start growing and getting bigger and bigger. Haven't divided yet. They don't divide until they leave the ovary, but they are growing and getting bigger. They will gather supporting cells, those granulosa cells around them, and they will go from these little primordial follicles. They'll start growing into larger structures like you see right here. So there's our oocyte, and then there's all those granulosa cells forming a ring around it. Once we have that ring of cells, this structure is referred to as a primary follicle. Those supporting cells are called thecal cells. We'll get to the granulosa cells in a moment. I apologize for misspeaking, jumping ahead of myself. The primary follicle is going to be the primary oocyte surrounded by a layer of thecal cells. These thecal cells are providing nutrition and hormonal support. Now, these primary follicles are going to take about 120 days to reach the next step. So they'll take a while, they'll be growing up. So if you do a section through an ovary, you'll see a couple dozen of these because every month we're activating about 6 to 10 of these. So we got a couple dozen primary follicles scattered throughout the ovary. And so we'll see all of these, that one, that one, that one, that one, cross edge of that one, cross edge of that one. And so they're all over the place. Now, a primary follicle, after about 4 months, will then develop into a secondary follicle, and that's where some of those thecal cells will now be called granulosa cells, because now they've created a new protein receptor that receives follicle-stimulating hormone from the anterior pituitary. And so let's draw this really quick. So let's assume that, let's say this is our ovary, and we've got our, so we've got, we'll pick this one as our primary oocyte. And so before anything else, this would be a primordial follicle. Once this starts to attract fecal cells, now they're going to form a nice little ring of fecal cells surrounding the structure. That would be our primary follicle. As this continues to develop, that primary follicle is going to get larger. It's going to have more and more fecal cells form around it. And as those fecal cells form around it, they'll form multiple layers. They will secrete a non-cellular layer around the egg. A non-cellular layer surrounding the egg is called the zona pellucida. And so it's spelled right here, the zona pellucida. It's a non-cellular protective layer around the egg, provides cushion. We're going to need that after ovulation. Now, the outer layer, the inner layer of those fecal cells are now going to turn on a gene to create FSH receptors, and those cells are now called granulosa cells. So the outer layer of cells are still fecal cells. The inner layer of green cells, those are now granulosa cells. Having built FSH receptors, they obviously now receive the FSH hormone and they respond to it. They will start growing much larger and they will start producing a little bit of estrogen and progesterone in response to that FSH signal. Now, the secondary follicle will take about 65 days, about 2 months for development. And so again, you will typically see a dozen or so secondary follicles scattered throughout our ovary. And so let's take a quick peek. And so now you can see this larger follicle with multiple layers of fecal and granulosa cells, and that blue haze surrounding the egg, that blue haze is the zona pellucida. And so that's our secondary follicle getting larger and larger and larger. We will then develop into what is called a tertiary follicle. This is our third level. The tertiary follicle is going to get bigger and bigger and bigger, and when it does, it's going to have more and more of those fecal cells surrounding the granulosa cells. Now there's going to become a division between the granulosa cells inside and the fecal cells outside. And that space is referred to as an antrum. A-N-T-R-U-M, an antrum. That's going to be that fluid-filled space between the inner granulosa cells and the outer fecal cells. This is going to allow the whole thing to balloon up and swell up rapidly, which is going to allow more elbow space for that egg to start growing much, much bigger. That inner ring of granulosa cells is referred to as the corona radiata. So the corona radiata is going to be that ring of cells surrounding the egg. Inside of that is that zona pellucida, that's the non-cellular layer. So that's what I've drawn in blue. And then the corona radiata is the green cells, those granulosa cells, surrounding the egg. Both of these are going to be very valuable once we ovulate and release that egg. And so that's where we can see this secondary follicle. There's our oocyte. There's that zona pellucida. So here's that corona radiata made of granulosa cells. Here's that large fluid-filled antrum. And then way out here, all the fecal cells that make the outer edge of this entire secondary follicle. I'm sorry, tertiary follicle, tertiary follicle. I know it says secondary, ignore that. Now, one of the things to bear in mind is those tertiary follicles, and you're going to have about 6 to 10 of those scattered throughout the ovary at each reproductive cycle. And so those tertiary follicles, when they start getting big enough, they will start producing competitive enzymes. They will secrete competitive enzymes that will float through the ovary and attack other tertiary follicles. It's competition. Part of that quality issue is I want only the best tertiary follicle to reach ovulation and kick the egg out. And so anyone that's not strong enough dies. And so that competitive enzyme is going to cause my 6 to 10 tertiary follicles to die down to the point of one winner. And that one winner is referred to as a Graafian follicle, also called a mature follicle. It'll look just like a tertiary, just bigger, but that's going to be the one winner. Any of the losers that died off are referred to as atretic follicles. They suffered atresia, fade away. All right. Now, we get to the point of ovulation. So now we got one big Graafian follicle. Let's make him bigger. So we've got our big oocyte surrounded by his zona pellucida. The corona radiata made of those granulosa cells, and then the theca externa, and this whole thing is one big Graafian follicle. Eventually, we're going to signal this whole thing to rupture the outer edge of the ovary, and the egg with its zona pellucida and corona radiata is going to be expelled. That's ovulation. Now, that's when that primary oocyte is going to divide to become a secondary oocyte and a polar body, and all of that is going to shed outward. That's going to leave behind this Graafian chamber. We just broke the surface of the ovary, so tiny little blood vessels on the surface are bleeding. This whole Graafian shell is now going to fill up with blood. And so we'll see this large blood-filled pocket form right after ovulation. And that structure is referred to as a corpus hemorrhagicum, a bloody body. In Greek, that's what it means, bloody body. And so that corpus hemorrhagicum is a blood-filled Graafian follicle. Very quickly, those external fecal cells will consume all the blood. They'll get big and puffy, and we'll now have a large yellow structure called the corpus luteum, big glandular structure. It's now going to start producing lots of estrogen and progesterone. We're making small amounts all the way back here at our secondary, tertiary, and Graafian stage. Now we're about to kick it up dramatically. Lots and lots of estrogen and progesterone. When you and I talk about the reproductive cycle, we'll see how that's valuable for helping to get a fresh stratum functionalis getting ready to grow in preparation for implantation. Well, that corpus luteum is only going to survive for about 2 weeks. It'll eventually fade away. It'll atrophy down to a little scar called a corpus albicans, white body. And so a little corpus albicans will be a little scar inside. And so what you're going to wind up seeing is this large structure is full of blood. The theca externa cells then digest and consume all that blood, and we get a big green puff or big yellow puffy structure. There's our corpus luteum. And after 2 weeks, the whole thing is going to just atrophy down until it finally forms just a little scar under the surface, and that's our corpus albicans scar. And that little scar is so small that it's really not going to interfere with any other eggs along the surface. It's not as bad as that ovarian cyst scar that's going to leave a big chunk. Those little albicans scars are negligible. They're not anything to worry about. There's your follicle development. And so that takes us through the various stages. And so we had our primordial follicle. That became activated, growing into a primary follicle, which grows into secondary, tertiary, Graafian follicle, one survivor. That one releases from the ovary the egg and fills with blood to become a corpus hemorrhagicum. The blood is consumed by thecal cells, which become very big, puffy glandular cells. The structure is now called the corpus luteum. After 2 weeks, this dies and atrophies down to a corpus albicans. And so you can see over on the right side here, there's our primordial follicles, primary follicles, we're getting a secondary, tertiary, there's our ovulation event, there's that puffy yellow corpus luteum. So again, you see this here with the primordials, primary, secondary, Graafian, ovulation, filling with blood, and then corpus luteum and corpus albicans. A lot of information. Take your time reading through the book, watch the videos, read this over and over again, chat with your classmates, talk to me, let me know if you have any questions. Good luck.